Good evening, everyone. I would like to introduce tonight Dr. Jacqueline Harris. She's from the Kennedy Creeder Institute, Johns Hopkins University, Assistant Professor of Neurology, Pediatrics, and Genetics. Dr. Clay Smith, also from Kennedy Krieger, Johns Hopkins University, and he's a neurodevelopmental physician.
Thank you, Dr. Esper, and thank you everybody for joining. Appreciate everyone coming to listen about Dr. Harris and I's recent publication titled Refining our Sleep Behavior and Adaptive Function in KAT6A syndrome. It was recently published in Brain Sciences and is publicly available. But we want to talk in a little more detail about our findings, and thank everyone for joining, and I want to thank our participants, the foundation, and Dr. Esper as well. So cognitive and behavioral phenotyping, phenotype being the observable characteristics that are attributable to genetic and environmental factors, but want to talk about sort of why we wanted to refine that phenotype in KAT6A syndrome, particularly in cognitive and behavioral realm. So I think that a deeper understanding can potentially lead to better insights into what individuals deal with or struggle with, and that insight could lead to potential environmental or pharmacological interventions that could modify outcomes for individuals. Notably, we also think that we need to have that refined understanding if we ever want to potentially modify with a pharmacotherapeutic or environmental intervention, to use that understanding as a potential metric for change. If a potential pharmaceutical company approached the FDA and thought that, maybe we could have a disease modifying therapeutic, then it's the burdens on them to prove that it actually modifies things. And so we need to have a good understanding to sell that. So to take a step back and talk about epigenetics in general, this is a figure from Dr. Svarner and Bjornson, who are in the Epigenetics clinic at Hopkins, one of their publications, that mentions or tries to portray the epigenetic or Mendelian disorders of epigenetic machinery, which is what we call these genes that modify other genes, and classify these things into things like a writer, an eraser, and a reader. And you won't see KAT6A syndrome actually included on this. An updated figure would, but KAT6B you may see in the writer realm here. And particularly KAT6A serves as a transcriptional coactivator, meaning it helps sort of recruit genes involved in making RNA from DNA or genetic material. And it also serves to acetylate a lysine residue on a particular histone. And the DNA is wrapped around these histones, and it needs to be compact and organized in each individual cell. So these nucleosomes, or these histones, are conformationally bound, and then the acetylation can change that conformation to make the DNA more readily accessible for things like transcription and making RNA and proteins. Again, another realm that we'll touch on just briefly here is something called long-term potentiation, which is it's our best model for how we remember things and learn, but it's basically how our neurons are reinforced after signaling. So long-term potentiation can be measured directly and can also be measured somewhat indirectly or by proxy. But these epigenetic disorders often have issues with long-term potentiation. Notably, there are some preclinical studies, meaning mouse models, where in Mendelian disorders of epigenetic machinery that that these things are potentially modifiable. So Rubinstein-Taybi, for example, there are some preclinical studies where drugs that manipulate histone acetylation and potentially augment that can improve long-term potentiation. And so that's sort of the moonshot for us, I suppose, is looking at these preclinical studies and how can we potentially modify cognition and behavior. So to go back to our study, looked at a few realms using some questionnaire-based information. And we looked at adaptive measures, how individuals function and in the home setting, how they function academically, how they function socially. And so we used a measure called the ABAS, and it reports conceptual, social, practical, and a composite or general adaptive composite score. And the mean scores for our cohort were in the 60 to 69 range. So for this particular questionnaire, that means that that's in less than or equal than 2.2% of the sample that they're comparing it to. So it's a representative population of the United States. So that's considered in the very low range. Now, the standard deviation or the each individual scores didn't necessarily all fall below this 70 threshold that we look at, but the averages did. The standard deviation did overlap into the low range above 70. Behavior, we had an instrument called the Child Behavior Checklist and Adult Behavior Checklist, and it reports things like internalizing, externalizing, depressive, anxious, attention deficit, hyperactivity problems. And none of the mean scores were in the borderline range, though again, standard deviations did sort of overlap into the borderline and clinical range. And we also looked at sleep. We used a different type of instrument that didn't have a normed or validated sample to compare or to make percentiles comparisons, but just gave us an idea about rates of issues in sleep. And there are really high rates of sleep maintenance issues, restlessness, bruxism or teeth grinding, enuresis or wetting the bed, and a need for medication. So this is one of the figures from the publication in regards to the adaptive and behavioral data. So we tried to separate the reports into what the mutation types were. Did this occur late in the gene? Did it occur early in the gene? And those were for ones that we think cut the gene off or truncate it. And then also essence or splice sites that just modify particular residues. So just broad strokes, looking at the general adaptive composite from the ABAS, all the mean scores were below that 70 threshold that we talked about, though the standard deviations overlap. And then just looking at the total problems. So this was the numbers are a different kind of scale here. Lower scores are quote unquote better in the behavioral reporting, and none of the average scores were in the borderline or clinical range. Here is the sleep data. These were reported just as sort of incidents. So how many occurred out of total reporting, and what was that percentile? So sleep quality seems to be an issue, with more than half reporting that their child wakes up in the middle of the night for prolonged periods of time or for multiple times, that grind their teeth, and 80% actually described their child as being just a restless sleeper. Enuresis or bedwetting, we cut that off for individuals reporting below 7 years of age, just to sort of make sure that we weren't reporting on normal developmental bedwetting, but 72% of those 7 and above reported that bedwetting was a regular occurrence. And then again, back to just general quality of sleep, 64% reported a need for medication pertaining to sleep. 72% reported significant drowsiness or sleepiness during the day. 68% sought advice or treatment, and 60% in general felt that their child had a sleep problem. So conclusions are that adaptive abilities were generally quite low, but there was a strong resilience in mood and behavior. Sleep is a domain where there's quite a bit of problem. And then we think that future directions could include a standardized cognitive battery to sort of sort out more details about how individuals with KAT6A think, what are their verbal intelligence quotients or IQs versus their nonverbal, and then looking at more quantitative measures for sleep. So things like actigraphy or watches that sort of measure general motion and things like that. We can extrapolate some data about quality and efficiency of sleep, or potentially more involved overnight sleep studies like polysomnography. I wanted to say thank you again to the KAT6A Foundation and to Dr. Esper, and of course all the wonderful participants. And I'll stop the screen share and look forward to chatting with you guys in a little Q&A section here.